Okay, so this is our recorded lecture on chapter 19, which is taxonomy, systematics, and phylogeny. Okay, so in terms of what we're going to cover in this chapter, the first part is on systematic biology. The second part talks about the three domain system, which we already kind of mentioned. I mean, we've mentioned this in Bio 111, and we actually mentioned it already in class. And then we'll talk a little bit more about phylogeny. Okay, so again, we've talked about taxonomy before when we talked about Darwin and natural selection, and we talked about some of the scientists that were around during his time, one being Linnaeus. And so we call really Linnaeus the sort of father of taxonomy. And so taxonomy is the branch of biology that's concerned with identifying, naming, and classifying organisms, right? So basically, we need to be able to classify organisms, right? Not only just identify them, but also classify them. And then that helps us to understand evolutionary relationships between different organisms. And this actually began a long time ago, right? So the Greeks and the Romans started naming organisms and organizing them into groups. And then we started to use these Latin names. And so we've continued using these Latin names, right? And basically the system that was set up by Linnaeus. I'll just say a little bit more about it in a second. This is just showing you, obviously, we're all aware of the many different types of organisms that exist, right? And so this is just showing you the sort of variety of different organisms that we have identified thus far. And there are many, many organisms or believed to be many organisms that haven't been identified, or many species that haven't been identified. Okay, and so as I said before, Linnaeus was the one who developed this system of binomial nomenclature, right? Binomial meaning two names. And the first word in that scientific name is the genus. Okay, the second word is the species, right? Or you can call it the specific epithet. But basically it refers to the species. Okay, so first word genus, which is capitalized, second word is species, and it's italicized. Okay, and so that's what this is telling you here. It says genus name alone can be used to refer to a group of related species, right? Which is fine. But if you want to give the specific name of that species, it's the genus first and then the species. Okay, and so some examples here, right? I mean, again, these plants look pretty similar, right? But you can see that these are two different species of lilies, right? So the scientific names, genus, Lilium, and this is the species Canadense, and then another, again, same genus, but different species, okay? All right, and so when we talk about classification and classification system, this is basically what we use, okay? And so this is going from most specific, right, to least specific, or this is least inclusive, meaning there are a fewer number of, right, this is only just one single species, to most inclusive, right, meaning you're going to have a lot of organisms that obviously fit underneath domain, okay? And so you can see the breakdown here. So a genus is one or more species, a family is one or more genera or genus, right? That's the plural form. In order, one or more families. Class, one or more orders. And so you can see how, again, when you get down to kingdom and domain, how they're going to include many, many, many more organisms than obviously genus does. Okay? And so you guys should know this order in which we classify things. All right, so just to give you an example of this, you can see here, right? So if you kind of look at these boxes, right? On the outermost box is domain eukarya, right? Both the mouse and the frog are eukaryotes. Kingdom Animalia, they're animals, right? Phylum Chordata, both of them fit into phylum Chordata, but they're going to differ from, class and then down, obviously, from there, okay? And so this mouse here, this house mouse, is class Mammalia. Genus Mus, right? Well, so sorry, class Mammalia, order Rodentia, right? Family Muridae, and then if you get down to the genus, it's Mus, and the species here is Musculus, right? So again, you can kind of see how these all fit in, okay? So it's a kind of a good way of kind of picturing this here, right? That you have this sort of biggest box as domain eukarya, and then all the other classifications or the other groups fit with inside. Here, again, if we're talking about this frog, the North American bullfrog, it's class Amphibia, right? Order Anura, family Ranidae, and then genus Rana, and species here, Caddisbayana. Okay, and Caddisbayana, I guess, right? So obviously, these Latin scientific names are not names that we use often, right? We mostly will go by these sort of common names, okay? But we should understand this classification system, again, in terms of the order, okay? I will not specifically ask you what the scientific name is of the house mouse, okay? All right. So it says, I already said this, the higher the category, the more inclusive, meaning has more organisms in it or more species in it. Organisms of the same domain have the same general characteristics in common. Obviously, members of the same species have very specific characteristics in common. Now, in terms of doing all of these classifications and identifying animals and putting them into these separate groups, right? When Linnaeus was around, most of what he relied on was anatomical traits, right? Behavioral traits and things like that. Now we really are using more molecular type of traits like DNA, protein sequence, and things like that. We'll talk a little bit more about that later on, but this DNA barcoding has to do with that, right? And so I want you guys to actually read this. I'm not really going to go over this now because I want you guys to read it and we can actually go over it together. So let me just show you in Moodle, okay? There is in your, if you go back to the 17, 18, 19 lecture materials, it's listed as DNA barcoding handout, okay? So sorry, it's kind of cut off there, but you'll see it. And it's here, okay? So I want you guys to read over this, just 2 pages here, and think about these questions, and we'll talk about it next time we meet. All right, so again, that's there for you. All right, so let's get out of here and go back to our PowerPoint. All right, and so that's what this is referring to. It's referring to that little, description about this DNA barcoding. And we actually did a DNA barcoding experiment in my research methods class like 2 years ago, and it went really well. So definitely something we'll talk a little bit about, okay? All right, so now let's move into talking about this 3-domain system. So we should definitely know the 3 domains and what makes up these 3 domains, okay? So it says sequencing of ribosomal RNA suggests that all organisms evolved in 3 distinct lineages. So again, this has changed. We didn't always have these 3 domains. And again, based on new data that we're able to obtain using RNA, using DNA, using proteins, like I said, we've come up with sort of some different way of categorizing, right? So this sort of field of taxonomy is ever changing, just so you guys know. So if you look at an older textbook, depending on how old it is, you might not see these organisms categorized into these 3 domains, okay? And so that's why it's mentioning this up here. All right, so the 3 domains, domain bacteria. And so if you think about what's in that domain bacteria, these are prokaryotes. They're unicellular organisms that reproduce asexually. They're bacteria, right? So you can have cyanobacteria, which can photosynthesize. However, there are many bacteria that are heterotrophic, right? So they don't produce their own food. These are particularly important in ecosystems, right? They are decomposers. Some bacteria are bad, we know that, cause disease, but we also have many bacteria, strains of bacteria that live within our bodies that keep us healthy, that help different processes like digestion and things like that. So anyway, we'll talk more about these guys in the next chapter. I'm sorry. Archaea, we probably don't talk about these guys much, but this is actually a totally separate domain. These are also prokaryotes. They're unicellular. They reproduce asexually. They pretty much live in extreme environments. They really don't affect us much, and so that's why we really don't talk about them very much, and we won't really talk about them very much. We'll sort of just identify the different types, and we'll leave it at that. Okay, again, that's kind of in the next chapter as well. It says the cell wall is diverse, but it's not the same as the bacterial cell wall. And so at one point, bacteria and archaea were kind of lumped together, but now with some of the molecular data that we have, we see that these guys are actually different, okay? And then the last domain eukarya. Now this domain is composed of both unicellular and multicellular organisms. However, they're all eukaryotes, right? So they have a membrane-bound nucleus. They have other membrane-bound organelles as well. And then you're going to start to see sexual reproduction. There's also some asexual reproduction as well, but but you don't see any sexual reproduction when you're talking about bacteria and archaea. And then you have the 4 kingdoms that are part of eukarya. So domain bacteria and domain archaea don't get broken down any further. However, eukarya does, right? So we're going to talk about all these different kingdoms in the next chapters, in the chapters ahead, right? So kingdom protista, fungi, plant, and animal. Okay, and so this is basically just showing you these 3 domains, right? We mentioned all this. So archaea, bacteria, and then eukarya. And you can see, obviously, there's a lot of diversity within domain eukarya. Okay, so like I said, the next couple chapters, we focus on, talking about what groups of organisms are part of domain eukarya. Okay, so this is basically what we just went through again in just sort of table form, comparing bacteria, archaea, and eukarya. Okay. All right, so again, you're expected to know those 3 main domains and what composes those 3 main domains. We move on to the last part of this chapter, which talks about phylogeny, okay? And so basically phylogeny is just the evolutionary history of a group. And again, we determine the phylogeny and make phylogenetic trees based on anatomical data, based on behavioral data, based on molecular data. And again, some of these phylogenetic trees have changed based on the fact that now we can use DNA and we can use protein sequence and things like that, okay? If you talk about systematics, it's the actual study of diversity of organisms using information from cellular to population level. So bottom line is it's the sort of branch of science that is responsible, I guess, for constructing these phylogenetic trees, okay? All right, so I already kind of said this. So we're going to look at a phylogenetic tree, and we just need to understand how to read a phylogenetic tree. Okay, nobody needs to memorize a phylogenetic tree, but we need to understand what it's showing you, right? So it says that if you look at the phylogenetic tree, and I'll show you a picture in a second, that each branching point is basically a divergence from a common ancestor, right? So there's some change here. That organism that exists at that branching point basically gives rise to 2 or more new groups there, okay? And when you look at the phylogenetic tree, you can basically see kind of 2 different types of traits, right? So something called ancestral traits, which these would be present in all members of the group and also present in the common ancestor, right? So this is a trait that you see in the common ancestor and you see in every other member of the group. Derived traits are traits that are present in some members of the group, but it's not present in the common ancestor, okay? So again, that's why it's called derived. So these are traits that came about later in the sort of evolutionary history of these organisms. Okay, so if we look at this and we kind of point out some ancestral traits and derived traits, we can, we sort of use this as an example here, okay? So actually, let me just bring in everything here and we'll talk about it, that way, okay? So if we can see here kind of the classification here, so class Mammalia, right? And then it's showing you these different orders and families here, okay? So all of these guys are mammals. And you can see this order Artiodactyla, okay? And family, okay, Cervidae, which is deer, okay? This family now is cattle, and so we can see kind of the difference here. We can also then look at the difference between primates and also this order, right? So this is order primates, this is order Artiodactyla, and then also some families within the order of primates, okay? And basically this is showing you these traits, right? And so you can look at ancestral versus derived, right? So if you're a member of this order, right, Artiodactyla, okay, you can see that you have, right, even-toed hooves here, right? Okay, so that's showing you this here. So here's your common ancestor mammary glands, right? That makes you a mammal, class Mammalia. If we move up here now and we look at one of these derived traits, which is this even-toed hooves, right? You can see that both the deer and the cattle have even-toed hooves, right? However, if you now go a little bit further, what's going to separate a deer and a cattle? One of the things is the horns, the type of horns, right? So, or whatever, horns or antlers. So deer have antlers, okay? And you can see here, but cattle have horns, okay? So this is the way that you read this, and I can put this on an exam and ask you questions about it. I don't expect anyone to memorize any of this, okay? So we should be able to, if we saw this, we should be able to pick out what is an ancestral trait and what is a derived trait, okay? And what sets these sort of families or these groups of organisms apart. Okay, again, if you look at, you know, within this order of primates, you can see, you know, monkeys, right, will have a tail. Apes don't have a tail, and they have this shoulder rotation, which is going to actually be more similar to the way, you know, a human sort of shoulder point. Okay, and that's why this is showing you here. Okay, so again, derived traits versus ancestral. You're going to basically see the ancestral trait being common in all of the sort of groups that you're talking about, where the derived are what's going to kind of set these different groups apart. Okay, so again, be able to interpret and read this. If we look at this phylogenetic tree, this was actually just another example of showing you a phylogenetic tree. This is actually showing you a phylogenetic tree of essentially of chordates. And you can see this common ancestor here. These guys, these lancelets, actually are not vertebrates, right? They don't have a vertebrae, you can see here. So everyone else over here from tuna up are vertebrates, okay? And so again, you can see common ancestor, and you can see all these different traits, these derived traits that set these groups apart, okay? So same thing. If I showed you this, we should be able to understand that this is showing you these traits that are, you know, unique to this branch here, okay? So if you talk about chimpanzees and dogs, they have hair and they have mammary glands, okay? A bird and a crocodile doesn't have hair and mammary glands, okay? So again, you can see what sets them apart. Okay, so to talk a little bit more about phylogeny, right? And we talked about this in lab too. So again, if you look at fossils and you look at the fossil record and you're trying to determine phylogeny, you know, evolutionary history based on the fossil record, it's kind of hard, right? It's difficult. You know, this is showing you a picture of an angiosperm, right? A flowering plant. And it was basically found during the Jurassic period, right? Or they identified it as being, you know, around during the Jurassic period. And that's how they basically figured out or determined that this is angiosperms. Sperms probably flourished during that time period, okay? But it's limited. This is all you have, right? You don't have any DNA information or anything like that. Some other kind of things that you, I guess, use in phylogeny, right? We talked about homologous structures versus analogous structures, right? And so the analogous structures come about via convergent evolution. We talked about this in the last chapter, okay? So I don't think we need to talk about this again. A homologous structure has the same evolutionary history, okay? Analogous structures may appear to have the same function, but they don't have common ancestry, okay? So again, they look similar because of adaptation to similar environments, but they are in fact not related evolutionarily speaking. All right, so again, if we're talking about phylogeny, we can use behavioral traits. We can use anatomical data like we talked about, right? And that's kind of the homologous and the analogous traits kind of fit in with this sort of anatomical or data. Behavioral traits like parental care, mating calls, etc. We talked about some of these things already. These are also used when identifying different groups of organisms. The big thing now is using molecular traits, okay? And so it says two species with similar base pair sequences are assumed to be closely related. Species that have differing base pair sequences are assumed to be distantly related. So when we talk about base pairs, we're talking about DNA sequence, right? And so the nitrogenous bases, right, whether we have an A, T, G, or C, right? We've all talked about this before in Bio 111. If we look at sequence of a particular part of a gene, we can compare that sequence between species, right? And that's what this is showing you, right? So this is just showing you 23 nucleotides, a segment of 23 nucleotides in a specific gene, and then comparing that sequence between cow, pig, this is the macaque monkey or whatever. This is just a, is that orangutan? No, a monkey. So anyway, that's just a typo there, just so you know. It's not some weird organism that you've never heard of before. So you can see where there is differences, right, in each position here. So if you look at the first, you know, nucleotide in a cow and a pig, it's a C. However, in a horse, it's a T, right? And then everyone else is a C. So you can basically look at these and you can see where you have differences among them, okay? So if you have a lot of nucleotide differences, it's going to indicate that those organisms are probably more distantly related. If you only have a few differences or they look almost pretty much similar, that's going to indicate that they're very similar in terms of evolutionary relationship. Okay. Other ways in other molecular data, and I kind of mentioned this as we went along, is protein comparisons. Immunological techniques, we did this in lab, right? So this is the degree of cross reactions used to judge relationship. That was using the antibodies that we used, and we looked to see if there was, this precipitate that formed. Okay, that's what that's mentioning. Also, you can look at amino acid sequence of different proteins. Okay, so again, if you see a similar sequence, it indicates a close relationship. If you see a more different sequence, it's going to indicate a more distant relationship. Okay, don't worry about this molecular clock here. This is basically, again, showing you that, again, these things have changed in the groupings, and the sort of relationship between these different groups has changed based on some of our molecular data, right? So the molecular data has really helped us determine the relationships and construct this phylogenetic tree particularly of the primate group. Oh, I thought I had a picture of, oh, sorry, I guess I don't have a picture of protein sequence. But anyway, similar to, let me just pull this back up, similar to what you might see with DNA sequence, you can do a similar thing where this would be a protein and you would look at different amino acids, okay? And so remember we talked about this in class, that we know that proteins are made up of amino acids. So if you looked at the difference between a mouse and a human, and you saw that in a particular protein they had maybe 5 amino acid differences, and then you looked at the cow versus the human and they maybe had 10 amino acid differences, that would be sort of your indication of how closely related things are. Okay, so just like you would do with DNA sequence, it's the same idea, but you would do it with protein sequence. Okay, and so that concludes our chapter 19 lecture. Make sure you review this and then before doing your quiz and your homework assignment. All right, thank you.